Hello, everyone, and welcome to the RNA Collaborative seminar series, today hosted by the University of Michigan. My name is Ljugman. I am the co-director of the Center for RNA Biomedicine, and I am very excited to introduce to you two very new faculty members here at the University of Michigan. They're so new that they haven't even started yet. But we have Sethu going to be our first speaker, and Chase are going to follow. So a brief background on Sethu Pitchiaya. He will be an assistant professor in the Department of Urology in about 8 hours, I believe, on July 1st. He got his undergraduate degree from Anna University in India. He then got his Ph.D. here at University of Michigan with Niels Walter, and he stayed in Niels' lab and worked on single molecule smart center that's set up in Niels' lab. And then he did a postdoc with Arul Chennaiyan here at the Department of Pathology, and he did a lot of nice work on link RNAs and rolling cancer. Then he stayed on in pathology, Department of Pathology, as a research investigator. And as I mentioned, he will start his assistant professorship in the Department of Urology tomorrow. Sethu has some nice awards. He got the Prostate Cancer Foundation Young Investigator Award. He has the AACR Bayer Prostate Cancer Research Fellowship. And he is the spotlight member of the RNA Society for this month, June 2021. So please check that out. It's a very nice story on Sethu. So Sethu's lab is really working on the fundamental mechanism of gene regulation. And today, he's going to tell us about the dynamic multivalent interactions that drive mammalian RNA regulation. So please take the floor, Sethu, or maybe I should say, take the screen, Sethu. So,
thanks, Mats. I'm hoping you're able to hear me okay, and I'm gonna share my screen now. And I'm hoping this looks good as well.
Yeah, it looks good.
Great. So yeah, thanks for that kind introduction, Mats. And I thank the organizers for actually giving me the opportunity to present some of the work that we've been doing over the past few months pertaining to how dynamics as well as multivalent interactions actually drive RNA regulation in mammalian systems. As I think Mats pointed out, I'm a Michigan homebrew, so you will see a lot of Michigan-inspired colors throughout the talk. So the flow of genetic information from DNA through RNA to proteins is intricately controlled via multiple steps, some of which can be called as the regulation of gene expression or gene regulation. The regulation of RNA itself is quite intricate and it's central to cellular homeostasis. For instance, if you were to take a very high-level picture of the life cycle of a messenger RNA, all the way from its transcription to its eventual degradation, what you may be able to appreciate is it's a multi-step process, number one, and number two, it spans multiple subcellular compartments. And we now know that errors at any of these steps actually leads to a wide variety of diseases, both genetic and oncogenic. And thanks to the ENCODE project, we actually now know that only a small fraction of the entire transcriptome, mammalian transcriptome, actually codes for proteins. The rest is actually extensively coding for what we are extensively has what is called as non-coding RNAs. And a lot of these non-coding transcripts are indeed functional, so the regulation by RNAs is by itself central to cellular homeostasis. And I'll just highlight a few RNAs here. For instance, long non-coding RNAs are now understood as master regulators of gene regulation at the transcriptional level. We have always known about the spliceosomal RNAs as well as the ribosomal RNAs, which actually are critical component of the gene regulation machinery and the gene expression machinery that forges life. And finally, we know also of small non-coding RNAs called microRNAs, which actually regulate gene expression post-transcriptionally via binding to 3' untranslated regions of messages. So if you were to take a step back and think about what are the potential factors that are important for RNA regulation, either by RNAs or of RNAs, three things come to my mind, and they are location, location, and location. Thanks to classical molecular biology, cell biology, and biochemistry, as well as contemporary genomics and microscopy techniques, we now know that the subcellular location of the RNA actually informs a lot on its fate as well as its function. For instance, sequences as well as structural elements embedded in RNAs actually can retain them in the nucleus. And for instance, these are aspects of long non-coding RNAs that mediate transcriptional regulation. On the other hand, transcripts can actually go to the cytosol, especially messenger RNAs, and localize at membrane-bound organelles, such as the endoplasmic reticulum or the mitochondria, where they actually make these proteins that are responsible for the functionality of the organelle itself, that is localized translation. So this process of RNA localization per se has been well characterized and well studied in polarized cell systems such as neurons and fibroblasts. However, ever since the time of Ramon Cajal, who drew these exquisite pictures of punctate structures in the nuclei of neurons, we now know that there are a wide variety of membrane-less organelles, for instance, P bodies and stress granules, as well as the nucleolus, which actually spatially regulate gene expression, and the mechanisms by which they act are only slowly being unraveled. And for the purposes of this talk, I will refer to these membrane-less organelles, that is, they don't have any membrane, as cellular bodies, phase-separated assemblies, biomolecular condensates, or membraneless organelles. And I sincerely apologize to any phase separation aficionados in the group. These cellular bodies are actually sustained in the cell via a process called phase separation, typically liquid-liquid phase separation, as oil droplets which persist in water. And this mechanism is actually quite emergent in the sense with the response to environmental cues. Proteins and RNAs can dynamically assemble into these assemblies, again, not predestined locations, but dynamically assemble into these assemblies, as shown in these two very nice videos. One on the left here that I'm pointing out to are P granules in embryos, and one on the right are examples of repeat expansion RNAs that actually form these clusters in the nuclei upon specific cues. At the molecular level, these cellular bodies are sustained by multivalent interaction, and multivalency can actually arise via protein-protein interaction or protein-RNA interaction. And what is slowly emerging is that the function as well as the property of proteins and RNAs in the dispersed phase as well as in the clustered phase could be widely distinct, and this distinction in functionality leads to a widespread heterogeneity in their function themselves. So if we were to think about what are the potential functions of these cellular bodies, they can possibly be highlighted into three categories. One is they can act as a reaction crucible, wherein you bring in reactants to enhance the chemistry of certain processes, which is evident in ribosomal RNA metabolism as found in the nucleolus. They could also act in a way by which proteins and RNAs are actually sequestered away from their site of function, which is seen in the case of TARC1 signaling and in response to stress. Finally, it could act as an organizational hub where proteins and RNAs, especially in multi-protein and multi-enzyme systems, such as metabolic pathways, could be spatially organized, which actually mediates these processes with high fidelity. This could also happen in terms of chromatin domains to regulate transcription. And the dysregulation of these cellular bodies leads to a wide variety of pathology, and it could happen in two ways. One, the formation of aberrant cellular bodies could actually lead to degeneration of the cell itself, and this has been seen in neurodegenerative diseases. Whereas on the other hand, it is possible that certain mutation, especially oncogenic ones, can actually lead to the dispersal of certain proteins such as SPOP in prostate cancer, which could actually drive the cancer itself. So formation as well as dispersion of cellular bodies in a dysregulated fashion can actually lead to a wide variety of pathologies themselves. So having given you an introduction of the dynamic evolution of cellular bodies and how they regulate RNA, again, in a dynamic fashion, I would actually take you through two distinct stories, and I would make them short at this point, but feel free to interact with me in the Discord channel, as well as feel free to ping me or email me later for more details. So in the first part of the talk, I'm going to talk to you about how multivalent proteins can rapidly and reversibly phase separate upon stress and actually modulate RNA metabolism. So cellular bodies actually mediate RNA regulation. Here, what I want to talk to you about are these RNA-bearing cellular bodies called P-bodies and stress granules, where P-bodies are a class of RNA granules that actually contain a lot of RNA metabolic enzymes and are present during cellular homeostasis. On the other hand, upon the induction of stress, and this could be in a variety of forms, including osmotic as well as the treatment of chemotherapeutic drugs, leads to the integrated stress response and the manifestation of what are called as stress granules, which are again amalgamations of RNAs and proteins, which actually contains a large number of RNA binding proteins and in fact eukaryotic initiation factors as well. And it has been found that the induction of stress also induces P-bodies. And my interest in P-bodies and stress granules comes from the cancer angle, where the tumor microenvironment is quite harsh, where it actually cells in the tumor microenvironment, especially cancer cells, face a wide variety of environmental stresses that leads to the upregulation of stress granules and P-bodies, which actually improves cellular fitness. And so it could actually contribute to cancer itself or oncogenesis. So having known this, we first asked the question of how do cellular bodies regulate RNAs during stress? So we modeled stress in the dish on these osteosarcoma cells that stably express fluorescently tagged DCP1A, or they actually, in this specific case, these were endogenous DCP1A, where DCP1A is a marker of P-bodies. We also co-stain via immunofluorescence for G3BP, a marker of stress granules. Upon induction of oxidative stress, and I'm just showing you one example of oxidative stress here, we've actually tried a variety of other stresses as well, such as heat shock, as well as drug-induced stresses. But in the condition of oxidative stress, we find that DCP1A and G3BP form these higher order assemblies only after 60 minutes of stress induction. On the other hand, when we treated cells with hypertonic stress, as in high salt, in this case 300 millimolar sodium, even within 2 minutes, you could see a rapid induction of DCP1A granules or DCP1A condensates, whereas G3BP was not forming such condensates, at least at 2 minutes, but at 60 minutes, you could see an induction of both DCP1A and G3BP consistent with stress response mechanisms kicking in. So as DCP1A condensation precedes G3BP condensation, especially upon hypertonic stress, we actually tested whether it was due to hyperosmolarity and actually treated the cells with actually sucrose as well as sorbitol and found it to be consistent with hypertonic stress. And due to the distinction between DCP1A assembly and G3BP assembly, genetically, we call this process HOPS, or hyperosmotic phase separation. And the acronym HOPS being very similar to an ingredient of a popular beverage is purely coincidental. So HOPS is actually a rapid process. Here's a video that shows it. Once you hit cells with high salt, you see that DCP1A assembles rapidly into these condensates, and upon the release of this stress, this is putting the cells back to isotonic conditions, a lot of these granules actually disappear. And this process actually is correlated with the physical compression and rarefaction of the cell, where upon treatment with hypertonic condition or hyperosmotic conditions, here what you're seeing here is a Z profile of cells actually vanishes, whereas upon rescuing cells, even within 2 minutes with isotonic media, the Z profile of the cells come back. So HOPS is rapid and reversible and is correlated with cell volume change, and all of this happens within seconds. So as DCP1A actually shows or exhibits HOPS, we want to find out what other proteins actually exhibit HOPS. So to this end, we actually performed high-throughput immunofluorescence for 108 different proteins, and the cells were either treated with isotonic condition, hyperosmotic conditions, or hypertonic conditions, or rescued with isotonic conditions after isotonic treatment. And upon analysis of this high-throughput data, we found that there were a group of proteins, as shown here in the top right part of this plot, that few, not all, actually exhibit HOPS. And when we were actually looking at these sequence elements in these proteins, a specific pattern emerged. For instance, a monomeric protein, here it's exemplified by GFP, does not exhibit HOPS. A dimeric protein, like AKT, actually also does not exhibit HOPS. A trimeric protein such as HSF1, though, starts exhibiting HOPS, much like DCP1A, which is also a trimer, and any multimer beyond a trimer, all the way until an octamer, such as PIGs, all of them exhibit HOPS, suggesting that any protein of valency greater than 2, that is a trimer or larger homomultimeric protein, exhibits HOPS. And this means that close to approximately 16% of the proteome has the capacity to exhibit HOPS. One of the hits from our data was CPSF6, or cleavage and polyurylation specific factor 6, which exhibited HOPS. And this protein is involved in the proper formation of 3' ends of transcripts, where CPSF6, as shown here, which I'm pointing out to, interacts with RNA polymerase II for the formation of 3' ends, proper formation of 3' ends. So now that CPSF6 forms these granules, we wanted to find out what can be the possible outcomes. It could either act as a reaction crucible, where actually transcription termination is enhanced, 3' end formation is enhanced, or it could actually act as a sequestration mechanism as CPSF6 is taken away from its site of action. To find this out, we actually performed metabolic labeling of RNA followed by bromouridine-mediated RNA sequencing in collaboration with Ljugman. And what he found was upon hypertonic treatment, there is widespread read-through of transcripts. And this actually is consistent with the data which was published by Joe and Stides' lab, where they also found that upon osmotic stress, you can actually see widespread read-through beyond canonical transcription end sites, and they call these transcripts as DOGs, or downstream of genes. So we wanted to identify the mechanism by which this happens. So we performed a modified chromatin immunoprecipitation followed by sequencing for CPSF6. And what we found was that upon hypertonic or hyperosmotic treatment, there is a widespread decrease in the occupancy of CPSF6 at transcription end sites, consistent with the sequestration mechanism, where under isotonic conditions, CPSFs are actually close to the gene bodies, mediating proper transcription termination and 3' end formation. Whereas upon hypertonic or hyperosmotic treatment, CPSF6 is actually sequestered away from its site of action, and this leads to transcription termination defects. So HOPS can potentially lead to a wide variety of regulatory paradigms. So we wanted to test what is the impact of hops on cellular fitness. To this end, we treated cells. Here is a cell that expresses GFP tag BCP1A, and we treated this with 1,6-hexanediol, an inhibitor that inhibits phase separation in a widespread manner. And we found that under isotonic conditions, these clusters disappear, and upon rescue with media that does not contain 1,6-hexanediol, you can see these clusters come back. On the other hand, if the treatment occurs in the presence of hypertonic or hyperosmotic conditions, cells tend to bleb. You can see accumulation of TCP1A at cell boundaries. And this actually does not get rescued even after washing off the drug and putting the cells back in isotonic media. So hops inhibition actually leads to impaired cell volume recovery itself. So put together, we arrive at a multi-scale model of hops and its potential implications, where at the cellular level, cell volumes change rapidly upon hyperosmotic treatment, which manifests as rapid and reversible phase separation of proteins at the subcellular level. At the molecular level, the proteins that exhibit hops actually have a valency of greater than 2, that is, trimers are larger. And at the atomistic level, we think hops is reliant on hydration and dehydration. And this process, again, with potential implications in cell biology in a widespread manner. Again, it could be involved in cell adhesion, invasion, migration, as expected in development as well as cancer metastasis. It could be a possible mechanism of action in mechanical stresses, as well as rapid changes in osmolarity as seen in organs such as the gut and in nephrons. In long time scales, that is, chronic stress could actually have widespread implications, both in a cell cycle where the cell volumes change in a much slower fashion, as well as in tumor biology, as well as in nephritic pathologies. With that, I will actually switch gears and jump to a second part of my talk. This will be pretty short, and in the spirit of this talk, being an RNA collaborative seminar, I'm going to present a lot of unpublished data, so feel free to give me all your feedback. And here we're going to talk about how micro-RISC assembles into higher order assemblies to promote RNA silencing. So microRNA-mediated gene silencing is a widespread evolutionary conserved mechanism where small RNAs, called microRNAs, guide the RNA-induced silencing complex, at the heart of which there is AGO, or organoid proteins, which recruits these proteins called GW182, which in turn recruits proteins that impacts deadenylation or translational repression. And microRNAs actually act, again, as in a widespread manner. Close to 70% of all messages are actually regulated by microRNAs, and any misregulation in the expression of microRNAs leads to, again, widespread pathologies. But one lingering question remains, in the sense, how do microRNAs find, bind, and repress their targets? It's estimated that there are about 200,000 copies or so of microRNAs and RISC complexes, whereas there is close to 1 million copies of messages, and sometimes the messages can have no or even 1 or 2 microRNA binding sites. So what is the stoichiometry of this interaction for robust silencing, and how do microRNAs actually find their targets? To understand this process, Niels Walter's lab actually developed an assay to actually look at RNAs, as in mRNAs and microRNAs, interacting in situ in living cells, where you can co-inject microRNAs messages, biotinolated phalloid and streptavidin, where the message itself is also biotinolated and labeled, and you can actually tether messages in the cells by this biotin phalloid to actin cytoskeleton, thereby increasing the visualization of microRNAs and messenger RNAs inside cells. And you can visualize multiple molecular species, as shown here in yellow, one where microRNAs and messages interact, green alone is where microRNAs don't interact with messages, and in red are where messages don't interact with microRNAs. And when Hui Li and Amir Jalihal in Nils's lab actually performed this assay on several different messages that contain different microRNA response elements, as in different number of microRNA response elements, they found two things. One, the interaction of microRNAs with the messages was highly dynamic, where a single microRNA could actually move away from a message, come back very close to it, and this could happen dynamically. So microRNAs reversibly bind messages. And the stability of this interaction, as shown here at the bottom in this heat map, where we are plotting the time unbound versus the time bound of the message against the microRNAs, the amount of MREs directly correlates with the stability. As you can see all the way to the right, the time bound of microRNAs with the messages increases with the increase in microRNA response elements. And it wasn't just that. It was also found that mRNAs end up clustering into higher order complexes upon actually interaction with microRNAs. And you can see here is a plot of a fraction of messages as monomeric or multimeric mRNAs. And you can see as the number of MREs increase, the clustering of the message also increases. And this data could actually be validated by single molecule fish as well, where a cell that expresses LET7 when introduced with messages that contain a LET7 bearing, MRE-bearing mRNA, you can see much lesser number of messages as shown here in panel C as expected for depression. And in panel D, you can also see that the intensity of these messages as compared to a control where the MREs are mutated is increased, suggesting that there is an MRE-dependent mRNA clustering. So to understand the molecular mechanism, we actually devised a system, an extract-based system, which was competent for RNA silencing as well as for translation. And here, when we actually made fluorescently tagged halo proteins or AGO proteins and put them on the surface of a microscope slide, we found that a majority of halo control proteins were actually monomeric, whereas a wide variety of wide fraction of AGO proteins were actually multimeric. So essentially, AGO2 ends up forming higher order assemblies in RNA silencing competent extracts. And this clustering actually is dependent on RNA. For instance, if you treat the cells with DNAs, nothing much happens. You largely see higher order clusters of AGO2. Once you treat them with RNAs, it actually fully reverses to being fully monomeric. So binding of AGO2 to RNAs is essential for this phenotype. If you actually mutate AGO2, especially at Y529 to a E, that is a phosphomimetic, which actually prevents the binding of microRNAs to AGO2, you also have this predominantly monomeric phenotype. So AGO2 actually forms higher order clusters in a microRNA and mRNA dependent fashion. Using these two pieces of data, we actually build a model where we find that microRNAs reversibly bind messages within clusters where there are multiple messages in the cluster itself that reduces the target search space for the microRNA, thereby leading to effective repression in a sub-stoichiometric fashion. And our data is consistent with previously published work from Ian McRae's group, where they found increased mRNA deadenylation and micro-risk condensation as well. And it was also consistent with our previous data, where we find that microRNAs as well as repressed messages actually localize to P bodies and outside of P bodies as well. With that, I will end my talk, and I would like to thank Arul Chinayan, my postdoctoral mentor, who's given me the opportunity to work on these distinct projects that are very different from what he does. And I would also like to immensely thank Niels Walter, my graduate student mentor, as well as my now close collaborator. And a big shout out to Amia Jalihal, a graduate student who plowed through much of this work along with me and also put up with me to some extent. With that, I will thank my funding bodies, and I would say the Pitchiaya lab, as Mats mentioned, will be opening in a few hours, and we are hiring. And thank you for your attention. I'll be happy to take any questions.
Thank you, Sethu. That was a great seminar, great talk. We're running short of time, but there's one question in the chat. and that refers to the CPCF6. Is it sufficient to knock that down to see transcriptional read-through?
That's a good question. We actually haven't done that assay per se, but it'll be a good one to test out.
And if you want to interact more with Sethu and Chase, you can go to the chat and get the information about how to stay on after the call, and they'll be willing to chat with you. So look in the chat for that information. Okay. So with that, let's move over to our second speaker, which is Chase Weidmann. He will be an assistant professor in Department of Biological Chemistry here at Michigan Medicine on September 1st. So Chase got his undergraduate degree from University of Rochester, and he did his PhD here at Michigan in Aaron Goldstrom's lab, where he looked at RNA stability, especially the role of familial and nanos proteins in regulating RNA stability. Then he left Michigan and went to North Carolina worked in Kevin Week's lab, did chemical probing of RNA structure. And he is currently a postdoc at Washington University with Ben Major, where he studies multiomics and RNA granules. And Chase is going to tell us about characterizing cellular RNA-protein interaction networks with chemical probes. Welcome, Chase.
Thanks, Mats. Can everybody hear me okay?
Yes.
Excellent. Yeah, so my name is Chase Weidmann. I'm currently staff scientist in the major laboratory at WashU in St. Louis right now, but as Mats mentioned, I'll be starting a new lab in September in the biological chemistry department at Michigan as a member of the Center for RNA Biomedicine and the Rogel Cancer Center as well. And sort of the ultimate goal of my research going forward is to try and identify these critical RNA-protein interaction networks that could become potential therapeutic targets. And ribonucleoprotein complexes, RNPs, especially long non-coding RNPs, represent this still largely untapped well of new potential therapeutic targets via multiple different strategies. And so I'm aiming to apply live cell chemical probing strategies to understand RNA-RNA, RNA-protein, and protein-protein interaction networks. I have limited time today, so sorry if people wanted to come and see some protein-protein mass spec stuff, but I'm going to focus on the RNA-centric technologies for looking at RNA structure and protein interaction networks today. But I am also applying varying cross-linking and mass spec approaches to understand the sort of protein-protein side of these networks as well. So I'll start with some examples, some published and some unpublished, of RNA function illuminated by RNA structure probing. And much of the structure probing I do relies on shape chemistry. And that stands for selective 2' hydroxyl acylation analyzed by primer extension. And in short, shape reagents like 5-NIA that I have highlighted here react with the 2' hydroxyl of flexible nucleotides. So if a nucleotide is base pair, it's largely unreactive to shape. And so if you probe an RNA with a shape reagent, you're essentially recording that RNA's structural state, marking flexible nucleotides with these chemical adducts. And the method I use for reading out these chemical adducts is called mutational profiling. In mutational profiling, you use modified reverse transcription conditions so that you can capture that RNA structure information by sort of coaxing the reverse transcriptase to introduce mutations and deletions right at the site of these shape reactivity. And then you can just read that information out by sequencing it. And I'll show you sort of what this data looks like in practice. So if you take, for instance, here's the U1 small nuclear RNA, whose atomic resolution structure is already known. But I'm going to represent this structure in two ways. Here on the left here in a linear fashion, where these arcs represent base pairs. Then on the right, a more familiar secondary structure of the RNA. And this is just to orient you to the type of data that I'll be showing. So when I extract this U1 RNA from cells and probe it with 5-NIA, we see sort of the expected reactivity patterns, right? So base paired stems are largely unreactive in black, whereas unpaired loops and single-stranded regions are moderately to highly reactive, labeled in yellow and red. Now, even if we didn't know the structure of U1 ahead of time, you can use this experimental shape data as added restraints when you're modeling base pairing with thermodynamic parameters. So depending on the number of potential pairings that might fit your data, you can assign pairing probabilities to all putative helices. And in this case, if you look at U1, there's a single highly favored structure that fits with the experimental data. And that's why these are all pretty much green arcs. And this does a pretty good job. You see it correctly identifies all of the known structures in U1 in a minimum free energy model. You'll see it also predicts this extra helix, which probably does form, but it only forms in the absence of proteins in the complex. And I've since applied shape to a handful of different non-coding RNAs and found unique features that are associated with different RNA behaviors. One of the link RNAs I looked at was this Slinker1, whose overexpression in melanoma cells is sufficient to promote metastatic behavior. Indeed, this was sort of discovered because its high expression is linked to poor melanoma outcomes. So this is my collaborator, Karen Schmidt, who originally found that this one fragment of Slinker1 appeared to be responsible for this effect. You could delete it, and in this case, this is an invasion assay. If you delete that fragment, you lose its invasion potential, and if you just express this one fragment of Slinker1, you get back essentially all of its invasion activity. And so I decided that it would be interesting to try and use shape probing to try and isolate what the functional element of this Slinker1 fragment is. So I extracted RNA from, in this case, patient-derived melanoma cells, and I probed with the 5-NIA shape reagent. And when I looked at this region of Slinker1, this is the part that's sufficient for the metastatic activity, I found sort of, well, maybe not surprisingly, it was pretty much just broadly reactive to shape. And it's not likely to be forming a lot of persistent and stable structure. If you compare this profile to U1 RNA from the same cells, which has a single stable structure, you can see that well-defined structure have these extended regions of low reactivity at stems and this sort of complementary hyper-reactivity at the loops. And maybe if you squint at this data on Slinker, you might be inclined to think that there's perhaps one stable structure in this region here. But indeed, when you try and model base pairing across this, it gives this very different looking to U1 pattern of these low probability couplings when compared, again, to U1. You'll also see a bunch of sets of overlapping arcs that aren't really compatible in the same structure, meaning that likely if this Slinker1 region is forming structure, it's likely to sample multiple different conformations. If you probe RNA with shape reagents in cells, you can actually spot regions of RNA that are resistant to reactivity because they're bound by proteins. And so I found significant protections from shape reactivity across this whole region, suggesting that potentially its inherent flexibility may be necessary to recruit a bigger RNP complex. And indeed, shape protections include this BRN3A protein binding site and both of these androgen receptor binding motifs. So my collaborator Karen and I, separately by EMSA and SPR, we found that this androgen receptor N-terminus actually preferentially binds to these unstructured pyrimidine-rich motifs, meaning that if the sequence is base paired, the androgen receptor won't bind to it. Thus, this sort of flexible region we found in this RNA is actually necessary to bring the androgen receptor in close proximity to this BRN3A binding site, forming this non-canonical transcription complex that ends up promoting metastasis through activating certain genomic targets. Now, because it's sort of reliant on the absence of base pairing in these AR binding motifs, we found you could inhibit the sort of metastatic potential of cells that are expressing Slinker1 using oligos complementary to the androgen receptor motifs. And I sort of bring this up as just as an example of how the sort of the lack of structure, not the presence of structure, in this case, drives the disease phenotype of this RNA. But I'll say that not all long non-coding RNAs behave in this way, as these sort of just floppy for protein binding platforms. So here's a different example. This is the MUNC link RNA, which controls differentiation during myogenesis. And here you see that the MUNC RNA appears to fold into these multiple distinct stable structures. You see a lot more motifs here that resemble what the U1 RNA looked like. And most of them are pretty well-defined. They have well-defined character throughout the whole RNA. And so you can use that structural information from shape to identify all of these different motifs, including a number of hotspots of potential protein binding from in-cell shape data. In particular, this CH4 motif had multiple instances of strong in-cell protections from shape reactivity. So I had this awesome graduate student collaborator, Roja, who recently defended her thesis. And she had a number of essentially established assays in mouse myoblasts that measure promyogenic function. And she just did this tour de force set of experiments where she tested how deletion or mutation of all of these shape identified structures affected phenotypic outcomes. And what was really interesting was that it seemed like different regulatory targets required different combinations of these MUNC structural motifs. So one of the targets, MyoD1, full activation of this gene's expression via MUNC required pretty much every one of those structural motifs to remain intact. And it was especially dependent on this CH1 and CH4 motif. But activation of two other myogenic targets, MyoG and MyH3, did not depend on all of the motifs. And MyoG could actually be activated even without the sort of, for all the other targets, usually critical CH4 domain. And interestingly, while these are all likely trans effects, because in these experiments, MUNC is being overexpressed exogenously, you don't see that mutant complexes are able to rescue each other in trans, meaning that CH1 and CH4 motifs have to be on the same RNA molecule in order to promote a response at the targets that depend on them. And in all likelihood, these domains are sort of intentionally bridging components together in a distinct complex, sort of unlike what Slinker might have been doing, i.e. just acting as sort of this floppy tether. So what I've brought up so far is you can see that structure probing, you can definitely identify important functional motifs. However, you can't really know just based on the structural information whether a flexible region or a highly structured region is more important than the other. So in both cases, Slinker 1 and MUNC, the important regions, one sort of unstructured and one more structured, are being engaged by proteins to exact their function. So I wanted to develop a probing method that just instead of doing it indirectly through shape, directly identified protein interaction networks with RNA. Because then I could theoretically more easily prioritize the motifs that are important, whether they were structured or not. So I had started by testing a handful of these potential RNA protein cross-linkers. I ultimately identified this chemical probe SDA, which has an amine reactive group and then a second photoactivatable group. The amine reactive group selectively reacts with flexible free amines, like those in lysine side chains. Lysines are one of the most prevalent side chains in RNA binding domains. And then upon photoactivation with, in this case, long wavelength UV, that diazurine group can rapidly cross-link to nucleotides in close proximity. And so because the presence of lysines in most RNA binding domains and the broad reactivity of diazurines with nucleotides, this sort of enabled capture of RNPs in a pretty much unbiased way. And I just want to stress that this is different than shape chemistry. A nucleotide just has to be in close proximity to the lysine amine to react. And its reactivity doesn't really depend on a nucleotide's pairing status. So SDA is cell permeable. Thus, it can be used to cross-link RNPs in live cells. And when you do cell lysis and protein digestion, you get these RNAs that have short peptide adducts on them. These adducts can be treated like any other RNA chemical modification. So again, I use altered reverse transcription conditions to encourage read-through of these peptide adducts, leaving diagnostic mutations and deletions in the product cDNA right at the site of protein cross-linking. And I didn't mention this before, but essentially because we're generating read-through events instead of termination events, multiple cross-linking events can be observed on single RNA molecules. So after you sequence the cDNA, you're going to get two types of information. First, there's going to be nucleotides with high mutation frequencies, and those are where proteins bind. Second, you're going to notice that some of these mutation events are independent, but some are going to be clearly linked. Mutations are going to co-occur on reads because multiple adducts formed on single molecules of RNA. And these events can result from a protein-protein interaction, the same protein bridging two locations together, or a coordinated assembly event. And you can imagine if you have different combinations of these correlated nucleotides, you can begin to describe complex protein interaction networks on RNA. And so this process essentially gives us RNP networks that are analyzed by mutational profiling or RNP-MaP. And so I'll show you again an example of what this information looks like, starting with an RNP of known structure. In this case, again, the U1 spliceosomal RNP. So on the left, again, is a linear model with the RNA stretched out, showing where the proteins bind to it. And then on the right is this sort of semi 2D, 3D model that represents the superstructure of the complex. And I found that nucleotides with the highest SDA reactivity correspond to known binding sites very well. And these RNP-MaP nucleotides are proximal to protein lysines, where SDA reacts. So that was good to see. Importantly, we get these correlations that reveal the interaction networks of the U1 RNA, including long distance interactions between the 70K protein and the SM protein complex. And additionally, the largest number of correlations seem to emanate from binding sites of this SM protein complex, which makes sense because it's responsible for initiating U1 RNP assembly. And this sort of highlights how RNP-MaP can identify the most critical parts of an RNP. And additionally, here's another example. RNP-MaP was able to identify conserved protein interaction networks on these RNase P and RMRP RNAs. These are both RNA components of RNase complexes that target different substrates. And so these sort of lines here represent a structure-based alignment because the sequences themselves aren't strongly conserved and the RNAs actually differ in length by about 80 nucleotides. Still, if you align them based on their shared RNA structure, RNP-MaP is able to clearly show that their protein interaction networks are essentially the same. They're totally shared. And that ends up being true. They actually bind to the same set of RNA binding proteins and fold into very similar shapes. And I find that, so these RNPs are defined by these protein interaction network hubs, where multiple strong correlations link together specific regions. And those shared network hubs actually define the substrate active sites, the specificity domains, and the sort of protein components that bridge together that specificity domain and active site. So essentially, in one RNP-MaP experiment, you can highlight all the important functional regions of an RNA of interest, irrespective of sequence conservation. I'll show you another example characterizing an even more complex long non-coding RNA. So here's RNP-MaP data for mouse and human exists. Long non-coding RNA, these are each about 20 kilobases long. And so again, you can see that not much of these RNAs are actually that well conserved, but in both mouse and human, these RNAs perform X chromosome inactivation, so they're accomplishing the same functions. But you can see that RNP-MaP is still able to highlight where the RNP networks are conserved, even when sequence is not. And it especially highlighted this region E here, whose sequence actually differs a lot between mouse and human. And what I found is that the proteins that actually recognize this E region are critical for formation of exist compartments in cells. So Sethu actually talked about these sort of RNP granules that form. Exist as one that does this. Without these RNA binding proteins, the cells sort of lose a significant number of these exist particles. And the particles that remain in the depleted cells are significantly less dense with exist RNA than those in wild type cells. And sort of interestingly, if you just take that chunk of the exist RNA, that E region, and you put it into an RNA reporter, it actually ends up in these huge foci that are similar to the compartments formed in the nucleus by native exist. So seemingly, this uniquely dense bound and interactive region identified by RNP-MaP is related to the localization of this RNA into foci. So interestingly, while I was developing RNP-MaP, the Lilly group at the University of Cambridge came up with this simplified strategy to enrich cross-linked RNP complexes with trizol, and they called it OOPS for orthogonal organic phase separation. And I wondered if this enrichment could enhance RNP-MaP signal. And so some of the work I've been doing recently to improve RNP-MaP beyond its first iteration, indeed, if you compare enriched cross-link fractions to free RNA fractions from OOPS, as opposed to treated and untreated samples in traditional RNP-MaP, this increases the signal about threefold, enabling higher sensitivity, fewer false positives, and a relaxed sequencing depth requirement for these correlation networks. And funnily enough, it makes the experiment easier to do and requires half the cellular input. So I'm pretty excited about that. So what I've shown you today is how some chemical probing technologies can be useful to pinpoint the functional regions of RNAs through their structure and protein interaction networks. And going forward, I'm looking to apply these technologies to other long non-coding RNA protein complexes that drive mechanisms of cancer metastasis. By integrating these chemical probing technologies, next-gen sequencing, and proteomics technologies, along with cell-based functional assays. And so with that, I will acknowledge my new faculty home, along with funding and collaborators that made all the work I showed today possible, as well as Ben Major's lab at WashU, which sort of doubled as a proteomics training central, as well as my general scientific home throughout the pandemic. And you'll also notice that Sethu and I talked to each other beforehand and inverted our Michigan colors in all the titles of our slides. So with that, I'll take any questions.
Well, thank you so much, Chase, for a wonderful seminar. Please go to the question and answer session to put in your questions, and we have a few here. So I start with the first one that came pretty early in the seminar, asking about, are you sure the base pairing is intra or inter interactions between bases based on the shape technique?
Based on the shape, no, there's not an exact way to know that other than just assuming that most structure is indeed local. Yeah, that's just the answer to that.
Okay. So the second one is, does SDA specifically react with RNA, or can also cross-links between protein-protein and protein-DNA occur? If it is specific, can you use it to identify proteins interacting with RNA bait?
Yeah, so it definitely is not specifically reacting with RNA, right? Like the sort of selective end of it is likely on the protein end with the NHS ester linking up with free amines. So definitely you'll get protein-protein, protein-DNA, anything that first thing reacts on that NHS ester, that diazerine is going to activate and cross-link into CH-OH and NH bonds. RNP-MaP sort of by nature of the way that you read it out, you're sort of selecting for the RNA specific information because you're using RT. But theoretically, and I've thought about different ways in which you can sort of use RNA as a bait, pull it out with oligos or enrich it some other way. And then depending on how you sort of release peptides into solution, you can get an idea of what are the sort of indirect contacts being made in a complex with your RNA, or the direct RNA binding domains of the proteins that recognize your RNA.
Okay, then we have, seems like diazerine has great potential. What is the time scale for labeling? How long is it applied on cells before harvesting them for follow-up assays?
Yeah. Oh, and then
And then
bioavailability.
the bioavailability in animals, is that known?
So for the cell-based assays, like originally I was doing it for 10 minutes of treatment, 10 minutes of cross-linking. That can probably be at least halved and could probably be shrunk even more, especially with the increase in signal from the enrichment method as well. You might be able to back off on the treatment even more. So the cells don't seem to care that much about it. And then the sort of cross-linking step, as soon as you sort of start hitting it with UV, the cross-linking happens and you've sort of frozen it in place. So really the sort of changes that can happen are probably happening when you add in the NHS ester portion before you cross-link. Bioavailability of diazurine, I would love to try this in so many different models, but I'm not sure about that. My guess is like, I don't have any good guesses, but I'd love to try something like that with somebody who knows more about testing bioavailability. I would probably start in something like C. elegans or worms, where it's like almost as though you're doing cell culture still.
All right, any more questions? Yeah, we got one more here. Fantastic talk. Can you explain how by RNP-MaP one can assess whether two RNAs are structurally conserved in a principled way rather than just looking literally, okay, it's moving up here, looking literally for similarities?
So I have like interesting ideas about, there's a lot of potential for doing things like this if the sort of SDA or whatever chemical probe signal for an RNP-MaP type experiment looks like, and whether or not you can figure out what one protein's binding profile by SDA looks like, because then you can sort of extrapolate it onto other RNAs. And then in that way, you can start looking for, oh, is this signature showing up on this RNA? Is this combination of different proteins showing up on this RNA in a similar way? You need more evidence yet to figure out whether that's sort of possible. But right now, looking for patterns in the correlation networks is probably the best way to do that for now.
Okay, one more just came up here. Can this method be applied in the reverse direction, i.e. to see what RNAs are bound to mutant RNA binding proteins?
I mean, yeah, it's just a cross-linker. It's just a matter of what method are you using to enrich the sort of protein? Then how do you get rid of the sort of, because you are going to get cross-links to other proteins as well. So you need to mitigate bridged interactions before you just search for, oh, what RNAs are associated with this, unless you sort of want what RNAs are associated with your mutant protein and the complex that it's in. Because the cross-linker is pretty short. It's like enabling the cross-linking at a 15 angstrom distance. So you might get indirect protein-protein, but the sort of level at which you can chain that is going to be limited to actual tightly bound complexes, I think.
Okay, we're coming up on the hour. So again, I would like to thank Sethu and Chase for wonderful seminars. And on behalf of the Center for RNA Biomedicine, we are so excited that you are coming to Michigan, starting here in Michigan. And we can't wait to see what's coming out of your labs when you get started on your own. So that's going to be very exciting. So I'm also very thankful for all the participants. So thank you all. If you want to interact with Sethu and Chase, you can go into the chat and look for how to connect to this little after talk, if you will, and interact with them. So please do that. And thank you very much for joining. And that concludes our RNA Collaborative for today. See you next time.